Welcome back, guys. After discussing about the contraction of the skeletal muscle as well as the cardiac muscle, now in this video we will be discussing about the contraction of smooth muscle. So first we will be discussing important points about the smooth muscle, how it is different from the cardiac muscle and skeletal muscle. Later we will be discussing how exactly the contraction of smooth muscle is going to happen. So first of all, let's see the basics, and later we will discuss more about the concepts. Okay. So let's begin smooth muscle. First of all, these smooth muscles, are they under your voluntary control? Okay, are they under your voluntary control? No, they are not voluntary. Okay, they are not voluntary in control. Okay, what is the shape of this? Smooth muscles. Smooth muscle, S for S, they are spindle shaped. Spindle shaped muscles. If you look at these muscles are going to look something like this, like a spindle, okay, like a spindles, okay, the nucleus in the center. They are the spindle shaped muscles which are grouped together, okay, like this. Now my question to you, they are not under your voluntary control. Normally your skeletal muscles are under your voluntary control. These skeletal muscles are innervated by the somatic motor neurons which are coming from the spinal cord. Okay, they are the alpha motor neurons which are causing the contraction of your muscles which are voluntary in nature. My question to you, these smooth muscles, they are not under your voluntary control. They are innervated by what? They are innervated by the autonomic nervous system. Okay, so they are not voluntary, they are innervated by autonomic nervous system, right? That is sympathetic or parasympathetic. Okay. Now question to you is, where are they present? Where is the location? Where exactly are they present? See, they are mainly present in your organs. They are the components of the organs. Components of organs. Which organs? See, they are present in the intestines. You know, your GIT is made up of 4 layers. There are muscles. There is a muscle layer called as a muscularis. There is mucosa, submucosa, muscularis and serosa. There is this layer called as a muscularis. Okay, so that muscularis. Okay, those muscles which are present in the muscularis. Are you able to contract your GIT? Are you able to do the contraction of your intestines? No, it's not possible because they are not under your voluntary control. So intestines that present, they helps in propulsion of the food. Okay, they help in the propulsion of the food. They are also present in your airways. So you know there is something called as a bronchoconstriction and bronchodilation. Okay, in asthma people there will be a reversible bronchoconstriction. So who is doing that bronchoconstriction? Can you do your bronchoconstriction? Can you make your bronchus to undergo constriction? It's not possible, right? So the smooth muscles around the bronchus is causing this constriction. So they are not under your voluntary control. Okay, so airways. The smooth muscles are present in your airways. What they will regulate? They will regulate resistance to airflow. Okay, they will regulate resistance to the airflow. But if you do bronchoconstriction, there will be resistance more for the airflow. If you do bronchodilation, there will be less resistance. Okay, if the smooth muscles are contracted, there will be bronchoconstriction. If the smooth muscles are relaxed, there will be bronchodilation. Okay, they are also present. The smooth muscles, they are present in many places, but for easy understanding, they are present in blood vessels, around the blood vessels. What they do in the blood vessels, again they are going to regulate the resistance to blood flow. Okay, if there is smooth muscle relaxation around the blood vessel, okay, the smooth muscles around the blood vessels. Okay, let me give you an example. Okay, smooth muscles around the blood vessels. What happened? For example, they are relaxed. Okay, they are relaxed. So what happened to the smooth muscle? The smooth muscle, what happened to the vessel? The vessel will undergo vasodilation. Okay, if there is vasodilation, what happened to the resistance to the blood flow? Resistance decreases. If resistance decreases, there will be more blood flow. Okay, there will be more blood flow. So, if you do vasodilation, there will be more blood flow. Okay, for that vasodilation purpose, what do you require? You require the smooth muscles to be relaxed. If the smooth muscles are relaxed means vasodilation, more blood flow. If the smooth muscles are constricted means, okay, the smooth muscles around your arteries. If the smooth muscle around your arteries are constricted means there will be less blood flow. Dilated means more blood flow. Simple. Okay. So, these smooth muscles are present, they are present around your uterus, okay, they are present around the bladder, okay. You can't contract your bladder, you try it, you can't contract your bladder. Maybe you can relax the external urethral sphincter, not the bladder contraction, it's not under your control, okay. You can't contract your uterus, it's not possible. So, the smooth muscles are not under your voluntary control, they are innervated by autonomic nervous system, not the somatic motor neurons, okay. Next, one more thing which I want you to know is, see, when I am discussing about the skeletal muscle contraction, okay, I have told you the skeletal muscles, they are innervated by the alpha motor neurons are there, these alpha motor neurons are going to release acetylcholine, and that acetylcholine is going to cause a depolarization in the muscle membrane. That depolarization in the muscle membrane, see the depolarization, electrical fluctuation, the depolarization in the muscle membrane is going to cause the calcium influx into the cell, that will cause the contraction of the muscle, we have seen there. But here in the smooth muscles, the action potentials are not always required, depolarization of the muscle membrane is not always required for the contraction, okay. So, let me write here, skeletal muscle requires action potentials or depolarization of muscle membrane. Or you can say muscle cell, depolarization of the muscle cell is required for what? For contraction. Okay, but what is the exception? Here the smooth muscles, okay, here the smooth muscles don't require, okay, don't require action potentials or depolarization for muscle contraction. Of course, you know, sometimes depolarization occurs, even smooth muscles, because of the depolarization or action potentials, even smooth muscle will contract. But what I am trying to say is, action potentials are not always required. There are other mechanisms by which the smooth muscles contract. Okay, that is the point. Now, see, if you look at the skeletal muscle contraction, the skeletal muscle contractions are going to be fast contractions. Okay, they are going to be fast contraction. Even cardiac muscle contractions are going to be sharp and fast contractions. But smooth muscle contractions are not the fast contractions. See, the skeletal muscle contractions are fast, okay, and these skeletal muscles will undergo fatigue very easily. But smooth muscles, they will undergo slow, sustained contractions. Okay, the contraction type, we will undergo slow, sustained contractions. Okay, look at the peristalsis. Okay, look at the peristalsis videos in the YouTube. See, they are going to be small, like slow and sustained, continuous for a long time. They are going to be slow contractions and sustained contractions, not the fast and sharp contractions. Okay, so slow and sustained. Here, this smooth muscle contraction, how it is different from the skeletal muscle contraction? Say, the skeletal muscle contraction is something thin filament mediated. Okay, the skeletal muscle contraction is something called as a thin filament mediated. Okay, thin filament mediated contractions. Why I am calling it as a thin filament mediated contraction? Because you know, on the actin there are which sites present? On the actin there are active sites present. Look here, active sites are present on the actin. Active sites are present. Now these active sites, are covered by a part of a thin filament called as a tropomyosin. There is something called as a tropomyosin. Okay, so this tropomyosin which is present on the thin filaments, this tropomyosin which is present on the thin filaments, it is preventing the contraction. So whenever the tropomyosin moves away, then contraction occurs. So it's something related to the thin filament, it is mediating the contraction. Okay, so if tropomyosin is there, no contraction. If tropomyosin moves away, then contraction will occur. We have seen that. But here, the smooth muscles, here in the smooth muscles, the most important point is, this is thick filament mediated contractions. The contractions of the smooth muscles are thick filament regulated. Okay, with big letters, I want to write here is thick filament, thick filament regulated. So why we are calling it as a thick filament regulated? So you all know what is a thick filament. The thick filament is nothing but myosin. Okay, so what is something happening with the myosin? Okay, what is something different with the myosin when compared to the skeletal muscles? Here, see, in the myosin, you know, there are myosin heads present. For example, if you look here, this is a heavy chain myosin. Now in the myosin there are heads present, these myosin heads present. Okay, now these myosin heads, there are heavy chains and light chains. Okay, there are heavy chains and light chains. Now see, these myosin heads, or I should say the myosin light chains which are present in these heads. So myosin light chain. Okay, they are modified when compared to the skeletal muscle. Here these myosin light chains, they are modified in such a way they will control when to contract, when not to contract. So it's the myosin light chains, they are modified to control contractions. So let me give additional info here itself, so that you will have a better understanding. See, okay, only this myosin light chain, the myosin light chain whatever was there, only if it is a phosphorylated, the phosphorylated myosin light chain will allow the contraction of smooth muscle. If there is no phosphorylation, means no contraction. So let me write here, only phosphorylated myosin light chains interact with actin. Okay, only in phosphorylated condition, then only these myosin light chains, they will interact with the actin. Okay, once the phosphorylation is completed, means now these myosin heads will go and bind with the active sites on actin, and that will cause the contraction. Okay, so who is doing this phosphorylation of this myosin light chain? Okay, who will do this process? So this phosphorylation is done with an enzyme called as myosin light chain kinase. Okay, myosin light chain kinase is going to do the phosphorylation, and that will going to cause the contraction. So contractions completed. Now I don't want any contractions. For example, because of some reason I just want to stop the contraction. My autonomic nervous system is going to stop the contractions. Want to stop the contraction. So what should happen? Now you have to dephosphorylate. Okay, phosphorylation is going to cause the contraction. So dephosphorylation. So with the help of dephosphorylation is going to occur with the help of an enzyme called as myosin light chain phosphatase. Okay, so that's going to cause relaxation. Okay, so these are the key points which I want you to know. So this is not something special to the skeletal muscles. This is not there in the skeletal muscles. There is no such thing as myosin light chain, phosphorylase, kinase. No, there is no such thing. But here something special inside the smooth muscles. See, these smooth muscles are designed in such a way that once there is phosphorylation of the myosin light chain, only then it will be brought to the particular configuration. Only then the myosin heads will brought back to the certain configuration, which will go and bind with the actin and will cause the contraction. Okay, so for proper cross bridge formation, for proper contraction, what do you require? You require the phosphorylation of myosin light chains. That's it. Okay, so let me write here how many forms of myosin light chain are there. Let me write myosin light chain. Okay, here also myosin light chain. Now, one form of myosin light chain, for example, look here, this form, it is phosphorylated. Inorganic phosphate I am adding here, for example, this is phosphorylated. Once you phosphorylate it, okay, from which form? This is an inactive form, for example, this form myosin light chain, here it is not phosphorylated. Now, what you have done it? You have phosphorylated with the help of myosin light chain kinase. Okay, now this myosin light chain kinase is going to do the phosphorylation, and who will do the dephosphorylation? That is myosin light chain phosphatase. Okay, now question is who is going to activate this myosin light chain kinase? We know, sir, myosin light chain kinase is going to do the myosin light chain phosphorylation. Now, this phosphorylated form is going to cause contraction. Okay, and this dephosphorylated form is going to be helping in relaxation. Okay, but my question is, when this myosin light chain kinase is going to be active, when it is doing the phosphorylation, so that requires something called as calcium-calmodulin complex. Okay, just for now, remember this one thing, there is something called as CACAM, that is calcium-calmodulin complex. So, this calcium-calmodulin complex, whenever it is there inside the cell, whenever the presence of calcium-calmodulin complex is there inside the cell, that is going to activate myosin light chain kinase, that is going to cause the myosin light chain phosphorylation, that is going to cause the contraction. Okay, that is going to cause the contraction. So, these are the basic points which I want you to know. Okay, how it is different, okay, from the skeletal muscles. Now, what else? See, in between these cells, for example, here is one smooth muscle cell. They are spindle in shape, but for easy understanding purpose I am just showing in the form of boxes. Here is the other here is the other smooth muscle cell. This is one smooth muscle cell, here is the other smooth muscle cell. Okay. Now, in between these two smooth muscle cells, there are certain connections present. So, these connections which I am showing here, the first connection, these are called as adherent junctions. Even in the topic of cellular connections or cellular junctions, we have seen that these adherent junctions are made up of 𝐒𝐶adherin, a protein called as 𝐒𝐶adherin. Okay. Now, here in between them, there are also other junctions present which are called as gap junctions. So, this fellow is called as a gap junction. Okay, gap junction. So, between the smooth muscle cells, what are present? Gap junctions. For example, if there are 10 cells which are in a series like this, 10 cells, one smooth muscle cell, second one, third one, fourth one, fifth one, something like that. In between these cells, what is there? There is this connection present. There are these gap junctions present, which will allow the substances to move from one cell to other cell. Okay, for example, because of the calcium, look, for example, let us say, here is one smooth muscle cell, here is the other smooth muscle cell, this is the third smooth muscle cell. Okay. Now, what happens is, for the contraction of even the smooth muscles, let me tell you, for the contraction of even the smooth muscles, you require the calcium. Okay, you require the calcium. So, for example, in one cell, calcium came. Okay, so this muscle is undergoing contraction. Okay, this muscle is undergoing contraction. But what happens is, within a fraction of millisecond, the calcium from one cell is going to go into the other cell. The calcium from this cell is going to go into the other cell. So, they are all going to contract at a single time. Because of the presence of these gap junctions, they all contract at a single time. Okay, and they will cause a perfect contraction at a single time because they are all connected. So, the connection is also called as a syncytium. They are all electrically connected, chemically connected because there are gap junctions. Okay, so these junctions are also, you can say, called as they are making a form of a syncytium, or you can say in another way, there are two types of smooth muscles. There are two types of smooth muscle cells. For example, one type here I have shown you, this is called as single unit smooth muscle cells. So what are the single unit smooth muscle cells? Here if one cell is contracting, other cell, they are all going to contract as a group. Contract as a group. Okay, so this is also called as syncytium. Okay, syncytium, there is connection. Okay, so this is one type, and the second type which I want you to know is there is something called as a multi-unit. Multi-unit smooth muscle cells. Okay, multi-unit smooth muscle cells. So this multi-unit smooth muscle cells means these cells are not going to contract as a group. They are going to contract as a individual cells. Okay, they are going to contract as a individual cells. So there are certain places in your body. Where you expect all the smooth muscle cells to be contracting at the same time. Okay, and there are certain places in your body where you expect the smooth muscle cells to contract individually. Okay, so if all the smooth muscles are contracting at the same time, that is called as single unit. Okay, that's going to be called as single unit smooth muscle cells. Okay, functionally they are syncytium, they are connected with each other with the help of gap junctions, and multi-unit smooth muscle cells, they will contract individually. Okay, individual contractions. Okay, so what are the examples for the multi-unit smooth muscle cells? They are present in your eye. In your eye, you know there are two smooth muscle cells, that is around the iris. Sphincter pupillae, dilator pupillae, you know it, right? So around the iris there are smooth muscle cells, that is sphincter pupillae and dilator pupillae. Okay, and ciliary body. Okay, so ciliary body muscles, they are also going to helpful for the accommodation of the lens. Okay, accommodation of the lens. Okay, sometimes your lens need to be flat and sometimes your lens need to be globular. Okay, for the proper focus on the retina. Okay, so these smooth muscles. Okay, these smooth muscles around the iris and the smooth muscles in the ciliary body. Okay, they are examples of multi-unit smooth muscles, and all the major smooth muscles, whatever you see, for example, in the intestines. Okay, the smooth muscles in the intestines, the respiratory system. Okay, smooth muscles in the intestine, respiratory system, uterus. Okay, they are all examples of single unit smooth muscle cells. Okay. Next, after this, what else I want you to know is we are seeing the differences, right? We are seeing the differences. Now, here, let's see some important points. How the smooth muscles are different from the skeletal muscles. In the skeletal muscles, we have seen there is a molecule. Okay, there is a molecule called as a troponin. So troponin, which is consisting of a troponin T, troponin I, troponin C, which binds with calcium, helps the movement of tropomyosin away. So such troponins are not present. Okay, troponins is absent in smooth muscle. Okay, so troponin is absent in the smooth muscle. We know it's a troponin which binds with the calcium. In the skeletal muscle, troponin is the one which binds with the calcium and is going to cause the movement of tropomyosin away. But here there is no troponin. Here there is no troponin. Okay, point number 1. Who else is not there? Okay, I used to remember it like TNT. TNT is absent. TNT. So N for nebulin. It's also absent in the smooth muscles. Next, other T for titin. You know, titin is a filament which connects the myosin with the Z line. Titin is the largest protein in the humans. Titin is like a spring, so it's called as a muscle spring. Titin is responsible for the maintenance of the elasticity of the skeletal muscles. So that titin is not here. Okay, so titin is also absent in smooth muscles. But my question to you, who binds with calcium? Who binds with calcium in the skeletal muscles? Skeletal muscles, it's the troponin C binds with the calcium. But who binds with the calcium in the smooth muscles? So when calcium comes, calcium and going and binding with. So who is that? Is there anyone who binds with calcium? Yes. So that I will be discussing now. So calcium in smooth muscle, once calcium come, how this calcium come, I will explain you. Once the calcium is available, calcium is going to bind with calmodulin and going to form calcium calmodulin complex. Okay, so calcium calmodulin complex we have studied somewhere. Where we have studied? So calcium calmodulin complex is going to activate MLK kinase, myosin light chain kinase. So that once the myosin light chain kinase, look here, once the myosin light chain kinase is activated, means that will cause phosphorylation of the myosin light chain. Once the myosin light chain is phosphorylated, now this myosin head is ready to go and bind with actin and causes the contraction. Okay, we'll come there. So what are the three important proteins which are absent? Troponins are absent, nebulin is absent, titin is absent. So these proteins are absent. But what about the tropomyosin? Tropomyosin, is it present? Tropomyosin is present. Its role is going to be different. Okay, its role is going to be different, but tropomyosin is present. Okay, in skeletal muscle, tropomyosin inhibits the actin-myosin interaction. But here in the smooth muscle, tropomyosin promotes actin-myosin interaction. Okay, next. There are certain proteins, okay, which are called as calponin and caldesmon. So who are these fellows? Calponin and caldesmon. See, think about the skeletal muscle, think about it. In skeletal muscle, it's the tropomyosin which covers the active sites. Tropomyosin covers the active sites and prevents the actin-myosin interaction. Normally, during resting condition, we don't want any actin-myosin interaction. Okay, so tropomyosin is inhibiting the actin-myosin interaction. But here in our smooth muscle, look, in our smooth muscle, it's a tropomyosin which promotes actin-myosin interaction. So who in the smooth muscle inhibits actin-myosin interaction? It is calponin and caldesmon. So right, they inhibits actin-myosin interaction. Actin-myosin interaction. Okay, so these are the points which I want you to know, the basics. Now, let's say calcium is it required for the contraction of smooth muscles? Of course, calcium is required. From where this calcium is coming? Okay, so question: calcium is coming from? Okay, calcium is coming from where this calcium is coming? You know, in the skeletal muscle, okay, the alpha motor neuron is going to release acetylcholine that is going to open the sodium channels. So sodium comes into the cell, the muscle membrane is getting depolarized. The depolarization will cause the opening of DHPR channels and RYR channels. So calcium will be released. That's the skeletal muscle story. But this is a smooth muscle. How the calcium is released into the cell? How the calcium is available for the cell to undergo contraction? For that purpose, you need to look at this, sir. Here is the smooth muscle cell. Okay. Now, look, even the smooth muscle cells, they are having the nerve supply. Okay, but the autonomic nerve supply, it's a autonomic nerve supply. Here, in the smooth muscles, there are no T tubules. Okay, no T tubules. T tubules are absent. Okay, the T tubules which are there in the skeletal muscles, T tubules are present in the myocardium, but T tubules are absent. MCQ, it's an MCQ. T tubules are not there. So just a little depressions are there. Okay, just a little depressions are there. Now here in these little depressions, there are these channels present. Okay, there are these channels present here. Let me actually show the neuron somewhere. So let me show the neuron here. Here is the neuron, autonomic nervous system neuron, ANS, maybe it's a sympathetic neuron, or maybe it's a parasympathetic neuron. Okay. Now, look, here I am showing you these are the calcium channels. Okay, here are the calcium channels. Now, when the muscle membrane, if it is a depolarized, I have told you, not every time depolarization is not required. Okay, not every time depolarization is required or action potential is required. For example, let's say if depolarization is happening, the muscle membrane is getting depolarized. Now, that depolarization is going to open these channels. So, for the first time, calcium is coming into the cell. So, from outside, the calcium, this depolarization is allowing the calcium to come in. Okay, the calcium came into the cell. Now, where this calcium is going to do? Okay, where this calcium is going to do? And from where this calcium is coming, first of all, so these channels which I have drawn here, these channels, these are L-type calcium channels. So, they are also called as DHPR channels, DHPR, dihydropyridine receptors. Okay, there are molecules called as the dihydropyridines. They will come and bind with these channels. The dihydropyridines will come and bind here. So, that's why these are also called as dihydropyridine receptors. They act like receptors for compounds like dihydropyridines. Okay. So, what are these channels called as? They are called as L-type calcium channels or dihydropyridine receptor calcium channels. They are long standing, okay, calcium channels. They are also called as CAV 1.2. Okay, that is voltage gated calcium channel 1.2. Where we have seen 1.1, voltage gated calcium channel 1.1 is present on the skeletal muscles and myocardium. Here it is CAV 1.2. Okay, it's voltage gated. Once the membrane is getting depolarized, the depolarization is going to open this channel, so calcium will come into the cell. So, where this calcium is going? Now, here in our smooth muscle, here I am showing you, there is the structure. So, if you have watched the video on the skeletal muscle, by this time you should have understood this is L-tubule. Okay, L-tubule. Okay, even here the L-tubules are not properly formed. Okay, so in the L-tubule, you know what are present? Calcium, lots and lots of calcium is present intracellularly, lots and lots of calcium is present in this L-tubule. Now look carefully, this calcium which came from outside, the calcium which came from outside is going to come and act on this channel which I am showing you here. So this channel which I am showing you here, this is called as ryanodine receptor RYR type 3. Okay, RYR type 3. RYR type 1 is present on skeletal muscles. RYR type 2 is present on the myocardium. RYR type 3 is present in the smooth muscles. They are present in the smooth muscle cells. So, now this calcium, it will come and stimulate this ryanodine receptor. The calcium which is coming from the outside, is stimulating the RYR channels. So, now once these RYR channels are stimulated because of the outside calcium, look what is going to happen. Now, the calcium is going to come out. So, for the first time, the calcium from the sarcoendoplasmic reticulum, actually this is the endoplasmic reticulum, SCR. So, the calcium is going to come out. So, this is how for the first time calcium is available in the cytoplasm. Now, the calcium is available. So, first calcium, this is the first calcium, this is the second calcium. So, the calcium which is coming from the outside is stimulating the RYR receptors to release the calcium from the sarcoendoplasmic reticulum. So, this is called as calcium induced calcium release. Okay, so calcium induced calcium release. So, first the extracellular calcium, this is the ECF calcium, don't forget it's the ECF calcium, okay, which came into the cell and that stimulates the release of the calcium from the sarcoendoplasmic reticulum. Just something similar to the myocardium. Even the myocardium, I have told you, the calcium which is coming from the outside, which is called as the essential calcium. In the myocardium topic, I have told you, the calcium which will come from the outside, that will stimulate the endoplasmic reticulum to release the calcium. Okay, there also I have discussed calcium induced calcium release is seen in myocardium, true. It is seen in the smooth muscles, also true. Okay, so this is one way you can get the calcium into the cell. And this calcium, what it will do? Ultimately, it is going to help in contraction. But is this the only way to get the calcium? No. Your smooth muscle cells, on their surface, there are these receptors present, maybe a G protein coupled receptor. Okay, it's a G protein coupled receptor. Okay, so even stimulation of this receptor, okay, stimulation of these receptors, what will happen is they are going to increase the secondary messengers inside the cell. So, stimulation of these receptors, which receptors I will tell you later. Okay, if you stimulate the receptors which are present on these cells, okay, or you can say it's a G protein coupled receptor. Okay, when you stimulate this, what happens is intracellular secondary messenger levels, that is IP3. Okay, intracellular IP3 levels are going to increase. IP3, inositol triphosphate. Okay, now this inositol triphosphate is also going to act on this sarcoendoplasmic reticulum. Even this IP3 can stimulate the release of calcium from this. Okay, so this calcium which I am showing you right here, now this calcium is released due to a secondary messenger called as inositol triphosphate. So remember, there are three types of secondary messengers: CAMP, CGMP, IP3. Now I am talking when you stimulate the receptors on the smooth muscles, for example like G protein coupled receptor, these receptors are going to increase the intracellular IP3 levels, inositol triphosphate levels. Now this increase in the inositol triphosphate will trigger the release of calcium. Okay, will trigger the release of calcium. So there is no action potential. I haven't discussed about the depolarization or action potential. In the beginning, here I have discussed about there is something depolarization, calcium comes in. But here I haven't discussed anything. There is no depolarization or no action potential. So IP3 is there, inositol triphosphate is coming. That inositol triphosphate is going to release the calcium from the sarcoendoplasmic reticulum. What this calcium is going to do? Calcium is going to bind with calmadelin. That calcium calmadelin complex is going to cause muscle contraction, phosphorylation of the myosin light chains. You know the stuff. Okay. So even without action potentials, the point which I want you to know, even without action potential, the calcium is getting released from the sarcoendoplasmic reticulum, and that can cause muscle contraction. Okay. So here the point which I want you to know is, okay, see there are certain drugs. For example, let's say there is a drug. Okay, there is a drug. That drug is coming and binding with this receptor, not a hormone. For example, not a hormone in your body. Normally hormones will come and bind with the G protein coupled receptors. Now you are using a drug. Okay, you are using a drug. This drug is coming and stimulating this G protein coupled receptor. Now what happens is the intracellular IP3 levels increase and calcium is going to be released. So what kind of interaction is this? In skeletal muscle, we have seen there is something called as electromechanical coupling. Electromechanical coupling. In the myocardium, we have discussed about electrochemical coupling. Here in the smooth muscles, even certain drugs will release the calcium out. Okay, these drugs are stimulating the receptor, so IP3 level increases, IP3 is going to release the calcium. So this is called as pharmacomechanical coupling. Because you know there are certain drugs which cause the smooth muscle contraction, there are certain drugs which cause the bronchoconstriction, there are certain drugs which cause the bronchodilation, there are certain drugs which will cause the uterine contraction, smooth muscle contractions. Okay, giving drug is going to cause the smooth muscle contraction. Okay, so what this drug is doing? That drug is stimulating these receptors, because of which intracellular IP3 levels increase. When IP3 level increases, that causes the smooth muscle contraction. So this is called as a pharmacomechanical coupling. Okay, pharmacomechanical coupling. So try to answer this. Pharmacomechanical coupling is seen with smooth muscle contraction. Now let me do integration with the pharmacology. Okay. Pharma integration. So what exactly is happening? There are certain drugs called as a dihydropyridine receptor, DHPR. Okay, dihydropyridine receptor calcium channel blockers. Okay, so if you are already a second year student, by this time you would have already known these drugs from your pharmacology. There are drugs called as dihydropyridine receptor calcium channel blockers. Can you give me any example? A famous drug, amlodipine, nicardipine. Okay, amlodipine, nicardipine. What they will do? See, they are in the names itself there. They are the calcium channel blockers. Okay, what exactly are they? They are calcium channel blockers. Which calcium channel blockers? They are L-type. Okay, CAV 1.2. So which calcium channel is getting blocked? Look, so these calcium channels which I am highlighting right now, so you already know, so these calcium channels which I am highlighting, see, these are Voltage gain into calcium channel CAV 1.2. Now if you use these drugs called as amlodipine or nicardipine, they are going to come and block these channels. These channels will be blocked. Once if you block those channels, so what happens? Do you think extracellular calcium will come into the cell? No. If the extracellular calcium is not coming in, do you think RYR channels will be stimulated? No. Do you think the calcium will be released? So do you think the calcium induced calcium release phenomena will occur? No. So what happens? Let's see. Now these drugs are going to inhibit calcium induced calcium release. Okay, they are going to decrease the calcium availability. They will decrease the calcium availability to the cell. Now without calcium, tell me what happens. Do you think smooth muscles will be contracted? No. Muscle contraction. Or actually, to be frank, you should say the muscles are getting less contracted or the muscles are getting relaxed. The muscles are getting relaxed. So when the muscles are relaxed, tell me, so the muscles which are around the smooth muscles, okay, the muscles which are around the smooth muscles are going to relax. So because the calcium availability is decreased, okay, the calcium availability is decreased, so the vascular smooth muscles are relaxed. Okay, if the vascular smooth muscles are relaxed, what happens? There will be vasodilation. Whenever there is vasodilation, that can decrease BP. Okay, that can decrease BP. So these drugs which are called as dihydropyridine receptor calcium channel blockers, examples amlodipine, nicardipine, they are going to inhibit L-type calcium channels. Okay, they are L-type calcium channel blockers. When the L-type calcium channels are blocked, the calcium is not coming from outside, so calcium induced calcium release is going to decrease, so calcium is not available. So without calcium, the smooth muscles are not going to contract, so there will be smooth muscle relaxation. Smooth muscle relaxation is going to cause vasodilation and decrease the BP. So they are used in the treatment of these dihydropyridine receptor calcium channels, they are used in the treatment of BP. And not only that, they are also used in the treatment of heart failure. Okay, they are also used in the treatment of heart failure because they are going to cause the relaxation of arterioles. Okay, they are going to cause the relaxation of arterioles. When the arterioles are relaxed, so what happens? The afterload decreases. Okay, afterload on the heart decrease. Afterload is a load on the heart. Afterload is the pressure required by the ventricle to generate, the pressure required by the ventricle to open the aortic valves. Right. So now in this condition, afterload is decreased because arterioles are dilated. So arterioles are dilated, so afterload will be decreased. So that can be used in the treatment of heart failure. You will understand this better in your pharmacology and your pathology. In your medicine, you will understand this better. As a first year student, it's a little hard, but just remember, calcium is required for smooth muscle contraction. Calcium 𝐤𝐵 contraction. If the calcium is not there, no contraction. If you decrease the calcium, the muscles will undergo relaxation. Okay. So next thing, what I want you to know is the concept, the main concept. So who is going to regulate the smooth muscle contraction? Regulation of smooth muscle contraction, or you can say tone of the smooth muscles. Okay, regulation of smooth muscle contraction at the tone of the smooth muscles. So number one is calcium. So number one agent is a calcium. Calcium means calcium, more means more contraction, less means less contraction. Okay, number one calcium. Second fellow is myosin light chain phosphorylation. More phosphorylation means more contraction, less phosphorylation means less contraction. Okay, so now calcium and myosin light chain phosphorylation, these are the ones who are going to regulate the tone of the smooth muscles. How much contraction of the smooth muscles? More contraction or less contraction? Contraction is happening, but more or less. Okay, so as we have already discussed, nitric oxide, autonomic nervous system. Okay, autonomic nervous system, or even other agents like nitric oxide, or like some other dihydropyridine receptor calcium channels, or like some other agents which are stimulating the receptors. How they are going to regulate this muscle contraction? Okay, let's make it as simple as possible. So look, to increase the calcium, I have told you, if you increase the calcium, more contraction of smooth muscle. If you increase the phosphorylation, more contraction. Look, If you increase, there will be more contraction. So, how they are going to be increased? How you increase the intracellular calcium levels? How you increase the myosin light chain phosphorylation? How? So, to increase the calcium or to increase phosphorylation, your autonomic nervous system or others, they work to increase the calcium, okay, and to increase the phosphorylation. So, these things, autonomic nervous system or any other agent, they work through something called as secondary messengers. You already know one secondary messenger, that is IP3. Okay, IP3, inositol triphosphate will release the calcium. Inositol triphosphate increases the calcium levels inside the cell. So, more calcium means more contraction, more tone. Okay, so your autonomic nervous system will discuss in detail, and other agents, for example like nitric oxide, they all work through secondary messengers. Okay, so what are the different secondary messengers inside the cell? Can you tell me what are the different secondary messengers? There are 3 secondary messengers. One is CAMP, okay, one is CAMP, CGMP and inositol triphosphate. Now see, these 2 secondary messengers, whenever they increase inside the cell, when you increase the CAMP levels or CGMP levels, what will happen? Look, the CAMP is going to inhibit myosin light chain kinase. Oh my god! So far what we have discussed, myosin light chain kinase is going to cause the phosphorylation of myosin light chains, and that's going to cause the contraction. But what is the CAMP is doing? Because of any reason, if CAMP levels increase inside the cell, the myosin light chain kinase is inhibited. So without myosin light chain kinase, what happens? So the CAMP is responsible for relaxation. Okay, that's going to cause relaxation of the smooth muscles. Okay, now what the CGMP is going to do? CGMP is going to activate something called as myosin light chain phosphatase. Okay, it's going to activate the myosin light chain phosphatase. You know what this myosin light chain phosphatase will do? Myosin light chain phosphatase, look here, look, this myosin light chain phosphatase, what it will do? It will remove this phosphate groups, phosphatase. So it will remove the phosphate group so that myosin light chain will become inactive. So that is going to cause relaxation. So let me write. So the CGMP, okay, it is also going to be responsible for relaxation. Okay, but what about this IP3? I have told you IP3 is the one going to increase the calcium levels. It increases the calcium levels inside the cell. When the calcium levels are increased, that's going to cause contraction. So tell me, which secondary messenger is going to be responsible for the contraction in the smooth muscles? In the smooth muscles, it is IP3. It increases the intracellular calcium levels, and that causes contraction. So again and again I am telling you, this CAMP, okay, let me just erase and let me keep here red color so that you will have a better understanding. Red color means inhibition. Okay, so the CAMP and CGMP, what are they doing? They are responsible for relaxation. IP3 is responsible for contraction. Okay, so whatever we have discussed, let's keep it in the form of a simple format so that it will be easy for you. How many types of myosin light chains are there? 2 forms, active form, inactive form. For example, say myosin light chain. Here also myosin light chain. So this is inactivated form, the myosin light chain, inactivated form, which is not involved in contraction. So to convert this myosin light chain into phosphorylated form, okay, so it's converted into phosphorylated form. Who is helping? Myosin light chain kinase. Okay, myosin light chain kinase is helping. Now when does this myosin light chain kinases are activated? You already know it. So whenever there is calcium calmadulin complex inside the cell, whenever there is calcium calmadulin complex inside the cell, that's going to activate myosin light chain kinase. That's going to convert normal myosin light chains into phosphorylated myosin light chains. These myosin light chains are responsible for contraction. Okay, you know it. But my question to you, so who is going to increase the intracellular levels of calcium so that it will go and bind with calmodulin? So first of all, calcium should be available so that it will go and bind with calmodulin. So who is going to increase the intracellular calcium levels? It is IP3. Okay, it is inositol triphosphate. Okay, inositol triphosphate. One secondary messenger complete. Now tell me, what is that secondary messenger? What is that secondary messenger who is going to inhibit these myosin light chain kinases? The myosin light chain kinases are inhibited by CAMP. Okay, intracellular CAMP. And now tell me, what is that enzyme who is going to convert phosphorylated myosin light chains into dephosphorylated form? This is the dephosphorylated. Okay, who is going to do that? That is myosin light chain phosphatase. Okay, that is myosin light chain phosphatase. So what this myosin light chain phosphatase will do? It converts, okay, phosphorylated form to dephosphorylated form. For our exams, what you should know is the CGMP, cyclic GMP, the secondary messenger. It's going to activate. It's going to activate myosin light chain phosphatase. So the CGMP activates myosin light chain phosphatase. Now what happens? You are going to have the dephosphorylated form that's going to helpful in the relaxation. Okay. So what I am trying to put into your mind is, sir, here there are the secondary messengers like CAMP, CGMP and IP3. Okay. So these secondary messengers, they are all involved in the maintenance of the contraction or the maintenance of the tone of the smooth muscles. Okay. So now for better understanding, let's put like this. Example. Have you ever heard about a hormone called as vasopressin? Vasopressin. The name itself is there, vasopressin. It causes contraction. Vasopressin, it causes vasopressin, vasoconstriction. Do you know how it's going to do that? See, this vasopressin is coming, for example, imagine this is the blood vessel. Vasopressin is coming from somewhere. Okay, vasopressin is coming, it's acting on the blood vessel. It is acting on its receptor. Vasopressin is going to act on its receptor. Okay, for example, let's say here is the blood vessel. Okay, this is the blood vessel. Now this blood vessel, it is surrounded by the smooth muscle. Here is the smooth muscle. Okay, let's put it something like this. Okay, this is a smooth muscle. On the smooth muscles, there is this receptor. Okay, there is this receptor. This is V1 receptor. Okay, you will study this in your pharmacology. So there is a receptor which is V1 receptor. Now who is going to come and bind here? Antidiuretic hormone or vasopressin? Antidiuretic hormone or vasopressin, both are same. Now this antidiuretic hormone or vasopressin is coming and stimulating the V1 receptor. So what happens when you stimulate the V1 receptor? Do you know what exactly happens? Now there is an enzyme that will be activated, phospholipase. Okay, sorry, phospholipase C will be activated. Because of the increase in the activity of the phospholipase C, finally the IP3 levels inside the cell increases. You know what this IP3 will do? The IP3 will stimulate, see, the IP3 will stimulate the sarcoendoplasmic reticulum to release calcium, So what that calcium is going to do? That calcium is going to cause the contraction. Okay, the calcium is going to cause contraction of the smooth muscle. When the smooth muscle is contracted, when the smooth muscle around the blood vessel, when it is contracted, that's going to cause vasoconstriction. So look, I have told you your autonomic nervous system are others. I have given a statement here, your autonomic nervous system are others. Okay, see, your autonomic nervous system, okay, are others. Who is the other? For example, look here, this hormone, antidiuretic hormone or vasopressin, what it is doing, it's acting on its receptor. When you are acting on its receptor, it is increasing the levels of IP3. So for the proper muscle tone, for the contraction of the smooth muscle, either you have to increase the calcium or you have to increase some secondary messenger. Okay, so these hormones, they increase the intracellular levels of IP3, that IP3 further increase the intracellular levels of calcium, and that's going to cause a contraction that will ultimately result in the effect vasoconstriction. Or you can say prostaglandins. One thing completed, vasopressin, business completed. Okay, one thing. Next, for example, let's say prostaglandins. See, if the prostaglandins, if they are acting on the cells, you know, prostaglandins, what are they? They are vasodilators. How they are going to be vasodilators? Because prostaglandins increases the intracellular levels of CAMP. Okay, prostaglandin increase CAMP. You know what the CAMP is going to do? Ultimately, CAMP inhibits myosin light chain kinase. When the myosin light chain kinase is inhibited, there is no phosphorylation of the myosin light chain. So, no phosphorylation means relaxation of the smooth muscle. When the smooth muscles are relaxed, tell me what happens? When the smooth muscles are relaxed around the blood vessel, when the smooth muscles are relaxed around the blood vessel, there will be vasodilation. So, what I am trying to put into your mind here is about the two hormones. One hormone is called as a vasopressin. What is it doing? Vasopressin increases the intracellular levels of IP3. What these prostaglandins are doing? The prostaglandins increase the intracellular levels of CAMP. That increase in the CAMP is going to ultimately cause the smooth muscle relaxation and vasodilation. The other one is going to cause a vasoconstriction, something like this. For example, let's say one more example, last and final thing. Let's say norepinephrine or epinephrine. This norepinephrine, epinephrine, what they will do? They are going to increase the intracellular level of IP3, you know. Okay, they increases the intracellular levels of IP3. When they are increasing the intracellular levels of IP3, that's going to cause contraction of the smooth muscles. That will cause vasoconstriction. You know, epinephrine is going to cause the vasoconstriction. How? It's going to increase the IP3 levels. So finally, the point which I want you to know is there are certain secondary messengers involved in the contraction of the smooth muscle. What are they? CAMP, CGMP causes relaxation. IP3 is going to cause contraction. IP3 releases calcium from the sarcoendoplasmic reticulum. For the contraction of the smooth muscle, no action potentials are required. Okay, for example, like hormone or a drug can simply cause the muscle contraction without even causing the depolarization, without even fluctuating the membrane voltage. Okay, so that's the point which I want you to know. Next, one more important thing was there. One more important thing is there for your exams. Have you ever heard about nitric oxide? Okay, let me add it. Even if it takes some time, not an issue. Nitric oxide. Okay. So, nitric oxide is a derivative of which amino acid? L-arginine. Okay, L-arginine. It's a derivative of L-arginine. Okay. Now, this nitric oxide, what it will do? See, nitric oxide is produced by the endothelial cells. Always endothelial cells. Okay, your endothelial cells produce nitric oxide. Okay. See, you all know nitric oxide. Nitrates are vasodilators. You have studied nitric oxide, nitroprusside, nitroglycerine, nitrates are the vasodilators. How? See, this nitric oxide, it is produced by the endothelial cells with the help of enzyme called as nitric oxide synthase. What this nitric oxide will do is, it is going to activate this nitric oxide, it is going to activate an enzyme called as guanylyl cyclase. Inside the cell, it is going to activate the guanylyl cyclase. Now, once the guanylyl cyclase is activated, this enzyme is going to convert, it is going to convert the GTP in your cell into CGMP. Okay. So, once the guanylyl cyclase is activated, the GTP inside your cell will be converted to a secondary messenger called as cyclic GMP or cyclic GMP. Now, you already know the story. What the cyclic GMP is going to do, I have already explained you. The cyclic GMP is going to activate myosin light chain phosphatase. So, this myosin light chain phosphatase, what it will do? It will cause dephosphorylation. Okay, it is going to cause a dephosphorylation. Once the myosin light chains are dephosphorylated, now they are not going to be involved in the contraction, that will cause relaxation. So, now you understood how nitric oxide is going to cause the relaxation of the smooth muscles. Okay, relaxation of smooth muscles. Once the smooth muscles are relaxed, you know, once the smooth muscles are relaxed around the blood vessels, once the smooth muscles around the blood vessels, once they are relaxed, what happens? There will be vasodilation. Okay, so from the beginning Okay, from the beginning of your MBBS, you are studying that nitric oxide, they are vasodilators. Nitric oxide are the vasodilators. Now tell me how they are acting as vasodilators, because nitric oxide is going to activate the intracellular cyclic GMP, or I should say, it's going to activate an enzyme called as guanylyl cyclase. Guanylyl cyclase increases the cGMP levels, and cGMP is going to cause the smooth muscle relaxation. Okay, cAMP is going to cause the smooth muscle relaxation, cGMP going to cause the smooth muscle relaxation, but only IP3 is going to cause the smooth muscle contraction. Okay, so based on this concept, I will add a few more drugs here. Okay, I will add a few more drugs, that is nitroglycerin. Okay, so if you take this nitroglycerin, what happens? Okay, if you take this nitroglycerin, in your body it is converted to, okay, in your body it is converted to nitric oxide. Now, this nitric oxide, what it will do? It's a vasodilator. You know the stuff? So, it's going to cause the venodilation. Venodilation means less blood will be coming to the heart, so preload decreases, heart, like the oxygen consumption by the heart decreases, so it can be used in the treatment of angina. Okay, so nitroglycerin, what it will do? In your body, the nitroglycerin will be converted into nitric oxide. You know the function of nitric oxide, it's a vasodilator. There are drugs like nitroprusside. Okay, nitroprusside. What this nitroprusside is going to do? Nitroprusside itself contains nitric oxide. So, contains, it acts like a donor of nitric oxide. Now, the moment you take nitroprusside into your body, this nitroprusside, this compound in its chemical structure, it's going to contain nitric oxide. So, it donates the nitric oxide to our cells. You know the story. Once the nitric oxide is there in the body, it's going to increase the cGMP levels, and cGMP is going to cause the smooth muscle relaxation. Okay, so just integration. Now, after this, what else you should know for your exams is there are certain G protein coupled receptors. Okay, see, for example, if this is one smooth muscle cell. Okay. Now, on the smooth muscle cells, there are different types of G protein coupled receptors. For example, see, this is one receptor, G protein coupled receptor. Now, inside this, they are attached with, okay, subunits. Okay, alpha subunit, beta subunit. Okay, the subunits. Here, it is attached with GS. Okay, alpha subunit, it is a G stimulatory type of receptor. It's a G stimulatory type of a G protein. Okay, it's a G stimulatory type of receptor. Now, there is a different receptor. From your pharmacology, there is a total different type of receptor. It's a G protein receptor again, but inside it is attached with GQ subunit, GQ. And there is a total different type of G protein receptor. Inside it is attached with G inhibitory, GI. Okay, GI subunit. So, what they will do? For example, if you stimulate this G stimulatory receptor, there is a drug which is stimulating this G stimulatory receptor. There is a hormone which is stimulating this G stimulatory GS receptor. Okay, it's a G protein coupled receptor. Inside it is attached with G stimulatory subunit. Okay, so what happens? Look, when you stimulate this G proteins, for example, let's say G stimulatory subunit, if it is stimulated, if you stimulate this receptor which is attached with the G stimulatory subunit, okay, for example, the first one, you have stimulated it. So, what happens is, this subunit is going to activate an enzyme called as adenylyl cyclase. Now, once the adenylyl cyclase is activated, you know the story, ATP will be converted into cAMP. Okay, ATP will be converted into cAMP. Now, what the cAMP will do? It's going to cause relaxation. From this moment, you should say. So, if you stimulate a receptor which contains G stimulatory subunit, which receptors are? Later I will explain you. In a minute I will explain you. If you stimulate any receptor which contains a domain, intracellular domain, G stimulatory domain, so that's going to activate intracellular adenylyl cyclase, which increases the intracellular cAMP levels. If you stimulate G inhibitory subunit, what happens? That G inhibitory subunit inhibits adenylyl cyclase. So, now the cAMP levels decreases. cAMP levels are decreasing. So, that will cause what? That will cause contraction. If CAMP is there, relaxation, CAMP is not there means contraction. Okay. Next, one more type, GQ. So what this GQ is going to do, it's going to activate an enzyme called as phospholipase C. Okay. Now what happens if the phospholipase C is activated means it increases the intracellular levels of IP3. That IP3 is going to be responsible for contraction, you already know it. Okay, it's going to cause contraction. So here what I have discussed is different subunits, okay, like G stimulatory subunit, G inhibitory subunit, GQ. What are they activating intracellularly, which enzymes, and what is the result? So simply in your exam they will tell you G stimulatory activation, G stimulatory receptor, G stimulatory subunit activation is going to cause muscle relaxation or muscle contraction, muscle relaxation. GQ causes muscle contraction. G inhibitory subunit causes muscle contraction. Okay, something like this. Now, you will have a doubt. So, which receptors contain G stimulatory as their domain? Which receptors contains GQ as their domain or subunit? Okay, so that I am going to explain you now. G protein systems. G protein systems. For example, say, you know sympathetic receptors. What are the sympathetic receptors? Alpha receptors and beta receptors. Alpha receptors are of two types, alpha 1, alpha 2. Beta receptors, there is beta 1 receptor, beta 2 receptor mainly, beta 3, beta others are there. Alpha 1, alpha 2, beta 1, beta 2. These are the sympathetic receptors. Now, this alpha 1 receptor, see for example, here is a alpha 1 receptor. Now, to this alpha 1 receptor, inside what is attached? GQ. GQ is attached. Now to this alpha 2 what is attached? G inhibitory is attached. Okay. So let me draw it here. So GQ and beta 1 G stimulatory, beta 2 G stimulatory. Okay. So stimulation of beta 1 receptors, you know the story. Once you stimulate the beta 1 receptors, GS is activated, G stimulatory subunit is activated, that is going to activate adenylyl cyclase. Adenylyl cyclase is going to cause muscle contraction. Okay, beta 1, this G stimulatory, what it will do? G stimulatory is going to activate the adenylyl cyclase, so intracellular CAMP levels increases, CAMP is going to cause a muscle relaxation, you already know the story. Okay, you already know the story. So, next, what are the muscarinic receptors? M1 muscarinic receptor, M2 muscarinic receptor and M3 muscarinic receptor. M1, it is attached with, inside the receptor, it is attached with GQ, M2 is attached with G inhibitory, and M3 is attached with GQ. Okay, 𝐶𝐞, okay, something like this. So, I have explained you which receptors are attached with, anyway it will come in your pharmacology, but for here, alpha 1 receptor stimulation, tell me what happens. So, if you stimulate alpha 1 receptors, GQ will be activated. Where alpha 1 receptors are present? Okay, let me give you more clarity. See, here is a blood vessel. Okay, here is a blood vessel. Now this this blood vessel is surrounded by smooth muscles. On the smooth muscles, do you know which receptors are there? Alpha 1 receptors are there. So here is the alpha 1 receptor. Look, this is the alpha 1 receptor. Now, who is going to come and bind with this alpha 1 receptor? Epinephrine will come and bind with this alpha 1 receptor. Now, intracellularly, alpha 1 receptors are attached with GQ subunit. You tell me, sir, here is GQ. Now, what this GQ is going to do? Tell me. Sir, GQ, you know what it will do? It is going to increase the IP3 levels. And IP3 is going to cause contraction of the smooth muscle. When the smooth muscle is contracted, once the smooth muscle is contracted, what happened to this blood vessel? The blood vessel will undergo vasoconstriction. Okay, it's going to cause vasoconstriction. So done. So this is the mechanism which I want you to put into your mind. So secondary messengers are involved, receptors are involved, the subunits are involved. Okay, something like this. So finally, at the end of the day, I just want to give you the clarity. I don't want to prolong much. So what is the mechanism of smooth muscle contraction? Okay, so I am talking now autonomic nervous system is activated. For example, now the autonomic nervous system is activated. Okay, so autonomic nervous system is activated, and you know the story. The calcium will first come into the cell. Voltage gated calcium channels, you know, from the voltage gated calcium channels, calcium is coming into the cell. This calcium will stimulate, okay, the RYR receptors, okay, ranodine receptors to release more calcium from the sarcoendoplasmic reticulum. Okay, so finally, who came into the cell? Calcium is available, you know, because of the IP3, calcium is available, okay, or because of calcium induced calcium release phenomena, calcium is available. So finally, calcium is available. Now, what this calcium will do? Once calcium comes into the cell, what it will do? I have already told you. Once calcium comes, calcium will bind with calmodulin. So this is a protein. Calmodulin is a calcium binding protein. It's always there. It's always there inside the smooth muscles. Calmodulin. So this calcium calmodulin complex. Okay, what it will do? This calcium calmodulin complex is going to act on, or it's going to cause the myosin light chain kinases to be active. Myosin light chain kinase to be active. So once this myosin light chain kinases are activated, now tell me what they will do. Myosin light chain kinases causes the phosphorylation of myosin light chain. Once the myosin light chains are phosphorylated, now the myosin heads comes to a conformation, means a conformational change is going to happen. Now once this myosin light chains are phosphorylated, so till now the myosin heads were like this. Okay, the myosin heads were like this. Now once the myosin light chains are phosphorylated, now the myosin heads, now they are active, now they will go and bind with the active sites on actin and will cause the muscle contraction. Okay, previously how they are like this, the myosin heads, they are not active, they are simply like this. Once they are phosphorylated, now they are going to come to a conformational change, now they are going to get to a proper position, so that will be helpful for the muscle contraction of the smooth muscles. Okay, so myosin heads come to a conformation, a better conformation, and not only that, the ATPase activity in this myosin heads, normally muscle contraction is an active process, so you require the ATP. So the ATPase activity will also be increased because of this phosphorylation. Because of this phosphorylation, now the ATPase activity increases. Previously ATP is not able to be broken down, so there is no muscle contraction. Now the ATPase activity will also increase because of this phosphorylation. Okay, not only this, now this calcium calmodulin complex on one side it is doing activation of myosin light chain kinase. Okay, now what else it will do is, this complex are going to activate other kinases. Okay, so some other kinases, not that important. What these kinases will do is, this kinases, wherever you see the word kinase, what they will do, they will cause phosphorylation. Okay, so phosphorylation of what? Two proteins called as calponin and caldesmon. Okay, so what exactly is this calponin and caldesmon? I have explained you. They are inhibiting the interaction between actin and myosin. Okay, they are inhibiting the interaction between actin and myosin. Now, what this calcium calmodulin complex is doing, it is helping in the muscle contraction. So, it is activating some other kinases. So, they will do phosphorylation of this calponin and caldesmon. So, once these are phosphorylated, now they are not going to prevent. So, it prevents. Okay, once you phosphorylate them, what happens? Now there is prevention of inhibition. Okay, there is a prevention of inhibition of actin and myosin. Okay, there is prevention of inhibition of actin and myosin. Now guys, listen, this calponin and caldesmon, what they are doing previously, they are inhibiting the interaction between actin and myosin. Once they are phosphorylated, okay, now they are not going to inhibit. So there is a prevention of inhibition between actin and myosin. Okay, so these are some points which I want you to know. Finally, one more thing, say if you look at the smooth muscle, if you look at the smooth muscle like this, okay, now in a proper skeletal muscle, say in a proper skeletal muscle, there is a proper arrangement of actin, myosin, actin, myosin, actin, myosin, so that there will be I band, A band, isotropic band, anisotropic band, which is giving something called as a striated appearance. Okay, the actin myosin filaments are regularly arranged, actin myosin, actin myosin, actin myosin, so they are regularly arranged, so they are giving a proper striated appearance. So here in a smooth muscle, one point I want you to know is there is no proper arrangement of actin and myosin. For example, see here, these are actin filaments like this. Here are all the thin filaments, actin filaments like this. Now here are the myosin filaments which are present in between them. Okay, here are the myosin filaments. Here the orientation of the actin and myosin will be different. Here, for example, let's say here they are like this. Okay. So the actin myosin, they are not properly arranged. Okay, and that's also a difference between skeletal muscle as well as smooth muscle. Okay, so with this we have completed the whole topic, about the smooth muscle contraction. Once the calcium is available, you know it. See, the myosin head will go and bind with the actin and the cross bridge formation and the sliding filament theory. That is common. That's common between skeletal muscle and smooth muscle. The cross bridge cycle. Okay, that's common. Okay, but what is difference? I have already explained you. Okay, there is something called as myosin light chain phosphorylation which is required for the smooth muscle contraction. Okay, so that's why this is called as thick filament mediated contraction. But skeletal muscle contraction is thin filament mediated, it is a tropomyosin, okay, which is regulating the contraction of skeletal muscle, which is regulating the interaction between actin and myosin. Okay, so with this, all the important points whatever were needed for your exam, all are completed. Hope the video is helpful. Thank you.